Hello and welcome to IB Biology Notes with Dr. Kimmo. This is A2.3 viruses. This chapter is higher level only. What are viruses? Well, we can call them non-cellular agents because they do not consist of cells, but they still do things. They need a host cell to reproduce, and that cell usually gets harmed in the process, which makes viruses obligate parasites. Obligate meaning that they have no choice, this is their only way to reproduce, and parasites because the host cell gets harmed. They do not share common ancestry, which means there's no one single ancestor for all viruses. Some of the similarities between viruses seem to be due to convergent evolution. Common features of viruses include that they are very small, usually about 20 to 300 nanometers in size. They are assembled from their constituent parts. They do not grow like organisms do. They use RNA or DNA with the same universal code that organisms do, and they have very few genes. They are usually related to infection of the host cell, to replication within the host cell, and to lysis of the host cell. Lysis is when the host cell bursts open and releases the viruses. They have a protein coat called capsid, and they have no cytoplasm. Some viruses also have a viral envelope with glycoproteins. Let's look at enveloped versus non-enveloped viruses. Enveloped viruses have an envelope that surrounds their capsid. And this envelope is a lipid bilayer made from the cell membrane of a previous host cell. Such viruses make their own glycoproteins, which they insert into the membrane. And non-enveloped viruses don't have an envelope, but they still have genetic material and a capsid. Viruses come in many different shapes and sizes. Here we have, for example, the tiny polio virus, bottom right, the huge Ebola virus, left. Top row, you have the herpes virus, the measles virus, HIV, and the SARS virus, which is a coronavirus, just like the one that causes COVID-19. This is an atomic model of the virus that causes COVID-19. Each blob is one atom. Dark blue is for the envelope. Red is for one of the proteins embedded in the membrane. Green is another one of those proteins. And light blue is a third one, a very special one called the spike protein, which the virus uses to interact with the host cell. Orange is glucose, which makes sense if you remember that these proteins are glycoproteins, proteins with carbohydrates on top. Viruses use RNA or DNA as their genetic material, but their genetic diversity does not end there. The genetic material can be single or double-stranded, SS or DS, and it can be circular or linear. This illustration shows some details that are not required for the IB, but that can nevertheless be useful. Number 1. Some viruses have double-stranded DNA. It can simply be transcribed into mRNA, and the mRNA is then used for translation. Some viruses have single-stranded DNA, which first needs to be replicated to produce double-stranded DNA, which is then used for transcription into mRNA. Number 3. Some viruses have double-stranded RNA, and one of those strands can then be used as mRNA. Number 4. Some viruses have a single RNA strand, which can be used directly as mRNA. Number 5. Some viruses have an RNA minus strand. It's the complementary strand of the final mRNA. So it first needs to be replicated to produce the mRNA, which can then be used for translation. And finally, retroviruses have a single-stranded RNA, which is first turned into a DNA molecule, a double-stranded DNA with an enzyme called reverse transcriptase. And this double-stranded DNA is then used to transcribe the genetic information into mRNA, and that mRNA can be used for translation. Viruses can go through processes called the lytic cycle and the lysogenic cycle. This illustration shows a phage, which is a type of virus that infects bacteria. You have the lytic cycle on the left and the lysogenic cycle on the right. Let's start with the lytic cycle. First, we have attachment, when the phage attaches to the bacterium. Then we have DNA entry, when the DNA enters into the bacterium, and then circularizes. You see the circular DNA of the phage in the middle of the slide inside the bacterium. Then DNA is replicated and transcribed inside the bacterium. After that, proteins are synthesized inside the bacterium and then assembled into phages, and you can see all these new phages inside the bacterium. Finally, the cell lyses and the phages are released and they spread. The virus can switch between the lytic and the lysogenic cycle. The lysogenic cycle also requires that the phage attaches to the bacterial cell and that the DNA enters the cell and is circularized. However, after that, instead of the cell producing a lot of new viruses, the DNA of the phage is integrated into the bacterial chromosome, becoming a prophage. As the cell divides, it copies the prophage and transmits it to the daughter cells. At the top right, you can see a daughter cell with a prophage. You see the blue phage DNA inside the bacterial chromosome. During the lysogenic cycle, the virus is undetectable and does not cause any harm. So how did viruses evolve? Viruses cannot survive without cells, so they probably evolved after cells. Viruses use the same genetic code as cells, so they probably evolved from cells. The high diversity of viruses suggests multiple origins, and there are two main types of hypotheses. There are progressive hypothesis and regressive hypothesis. According to the progressive hypothesis, viruses evolved from cells that started to spread their genes to other cells. This is supported by the presence of so-called retrotransposons in modern eukaryotic cells. Retrotransposons are DNA sequences which are transcribed into RNA, and the RNA is translated into protein. These proteins include reverse transcriptase, and the proteins along with RNA is transported into the nucleus, where the RNA is reverse transcribed back into DNA. This DNA is then integrated into the genome, which results in new copies of retrotransposons in random locations in the genome. According to the regressive hypothesis, viruses evolved from cells that lost more and more components, eventually becoming entirely reliant on other cells. This is supported by a variation in self-reliance and complexity of both viruses and bacteria. For example, there are small and simple viruses like the poliovirus, 30 nanometers, 7,500 base pairs. And there are large and complex viruses like the virus, up to 750 nanometers and 1.2 million base pairs. There are also parasitic Rickettsia bacteria, which can be as small as 100 nanometers. That's way smaller than the large virus, for example. These bacteria grow and replicate only inside eukaryotic cytoplasm. The Rickettsia genome has similarities with that of mitochondria, which you may remember evolved through endosymbiosis, and Rickettsia cause diseases like spotted fever and typhus. The bacterium Chlamydia trachomatis is another well-known intracellular parasite. Viruses evolve fast because they have a very short generation time, they have a very high mutation rate, and they experience very strong selection pressure. Examples of viruses with rapid evolution include the influenza virus, HIV, and SARS-CoV-2. That's all for now. See you next time.